daily dose of honest science.
Hi and welcome back to Daily Dose of Honest Science. This is part 2 of actually episode 1 from season 2. And we're gonna deep dive into the molecular subtypes of Alzheimer's disease that was detected in CSF. And we're gonna discuss a lot and also what is the limitation of the study and what kind of take-home message can you take from it. Have a lot of fun and enjoy. And now we're gonna go to the results that they showed. And Corinna, you, I think you're the best in explaining subtype 1 and 2 actually.
I can give it a try. Well, their analysis of the protein samples and then comparing it with information they had in addition to all the different patients, as you have already pointed out, they identified a so-called subtype 1, which is the hyperplasticity type. What does that mean? Among the proteins that were regulated, and most of them upregulated, they found proteins that are associated with neurons, astrocytes, and different cells you find in the brain, which is good, I guess.
Yeah.
They did find proteins that are mainly associated with neuronal plasticity and processes at the synapse. And if you remember what I had explained at the beginning, what happens early in Alzheimer's disease, that I told you that normally there is a reduction in this plasticity. So in subtype 1, they found actually an increased plasticity, which is a little bit stunning, and I wouldn't have expected that. And then they went on and analyzed a little bit more the samples of this subtype, and they found out that the subtype 1 samples are associated with mutation in a molecule called TREM2. TREM2 is a surface molecule of microglia. Microglia are our immune cells in the brain, so to speak. And if this is mutated, this TREM2, these microglia can't do their job anymore. And or at least not as well. What one knows is that microglia, the immune cells of the brain, they are able to phagocytose something like the plaques and all kinds of things that are not good for our brain. So
they're like the cleaning agents
Yeah.
going through, right?
Yeah, they're sweeping the floor and taking the dirt with them. But if you steal their broom, they can't do that anymore. So these microglia are apparently in these patients not as active as they are in a normal brain. And so their assumption is, from their results, is that maybe due to the reduced activity of the microglia, you have a higher load of plaques. And this higher load of plaques is disturbing even more, and then the system tries to compensate in being very plastic and very active. So this doesn't mean that these patients remember their breakfast better than others. They probably don't. It's just that this remodeling activity is initiated apparently by even larger damage. So that's more or less what one can say to this hyperplasticity type 1, that there seems to be a lot of activity at the synapses, a lot of plasticity going on, most likely caused by this high load of plaques.
Because they cannot be efficiently removed anymore.
Exactly. So concerning subtype 2, there they found mainly proteins associated with the activation of the immune system in the brain. So actually the opposite of type 2, so to speak. So here we have very active microglia, and they might be so overactive that they remove even things that they shouldn't remove. So they see signals which are normally seen in the developing brain, where it's very common that you generate a couple of cells, they make contacts, and then during development, it's kind of like, it's called pruning. Synapses that are not needed are removed and others are strengthened and so on. So this information that's going on between the immune cells and the neurons when the nervous system is developing, these typical signals one sees in these subtype. And so one thinks that the overactive immune system is even removing connections and so on that should not be removed. And when they were looking at the imaging of these patients, if they were available, they could see that these patients were the ones with the smallest brain, so to speak, so the highest amount of shrinking of the brain, which we had explained in the beginning as well. So I guess everyone remembers that part.
Okay, so then I come to something different, so subtype 3, which is RNA dysregulation. So very shortly, what is RNA? So our body has inside the nucleus, of course, we have the DNA, right? We all know our DNA, our genetic information. But that is a master copy. And we don't want that our master copy gets destroyed. So the cell actually came up with a system to make a copy of our master copy. And this master copy is called RNA, ribonucleotides, nucleic acid. And there are some differences between DNA and RNA. I don't want to go much into it. Just that you understand, okay, it's about this copy that is happening in the brain. And so there they found like molecular pathways that were associated with cytoskeleton organization, axonal transport actually. Proteasome is when you degrade your proteins within the cell, right? And also protein folding, which means the proteins have a 3D structure and they need to, that's called folding. And it's mostly an active process. And this is somehow disrupted also in this subtype. And very prominently what they found were proteins associated with the processing of this RNA. So when the cell in the so-called transcription gets the RNA, and there are different RNAs available, it happens that we need to trim this copy. And this trimming is called splicing and some other factors. And they found out that here is the problem, that something is wrong, that they don't get trimmed as they should to be functional. And it has been shown, and I summarize now subtype 3 with that this kind of dysregulation already has been shown in other previous proteomics studies. So it's another confirmation for the subtype that RNA and the process of getting this one not correctly has an influence on the actual disease progression of Alzheimer's disease. So this is subtype 3. Now we come to subtype 4, which is the choroid plexus dysfunction. And you will see a picture where the choroid plexus is. And it's also bathing in CSF.
And it's the structure making the CSF.
That's the cool thing, right?
Exactly.
Yes. So when they looked at the proteins that are different, they found out that they have specific proteins measured that were specific to microglia again and other immune cells. And somehow they figured out that there were even other proteins that were highly expressed, which belonged to the lateral ventricle of the choroid plexus, right? And other proteins that they found all had biological processes tied to this region. And since it's a production of this, or involved in the production of CSF, it's very important also for the nutrition and lipid and protein transfer. And also is the blood CSF barrier. So this I also didn't know. It's really cool. And what was really interesting when they looked at the MRI scans was this type, specifically the fourth type, had a really large choroid plexus volume compared to control, but also to the other ones. And that has been shown in other studies. If you have an enlargement of this area, it's associated with inflammation and structure changes. And when they looked at the atrophy, actually, of these patients, it had the worst from 1, 3, and 5. And also they saw an increase of proteins who can build like cytokines, which also correlates to attracting more monocytes and T lymphocytes. So the immune cells are really highly upregulated in this subtype. And that's why they suggest that the subtype has an impaired clearance metabolism. Impaired clearance metabolism. Now we come to subtype 5, which is the blood barrier dysfunction. And this subtype was associated with other biological processes, and they found out like blood coagulation or B-cell mediated immunity, but also acute inflammatory responses. And what I took away from this one was that on MRI scans they saw microbleeding. So these patients had microbleeding. And that was something which you don't observe in the other subtypes. And the problem that they somehow said is like that these people kind of, through this very dysfunctional blood barrier, actually develop more of this AD because this, the plaques are somehow helping to loosen this tight regulated barrier. That was kind of interesting. So now we have all 5 subtypes, right? Were you surprised, Corinna, what they found out, or was some stuff you already knew and be like, yeah, well, that makes sense?
Well, what surprised me was that the results that they have, this subtyping due to the protein analysis, was going in line with the other results that they have from the patients, or the other data from the patients, that this subgrouping wasn't leading to a confusion, but it was kind of like they could confirm and put data together from other methods to investigate this disease, which do make sense. And this is really, really cool, because as you said, the data set is quite large that you get when you analyze these proteins and with the method applied. And that they were really able to make such a clear
distinguishing between,
yes.
Yeah. That's kind of rarity, yeah.
Yeah, that for me was the really surprising part and the really amazing part of that study, that in taking a sample from the cerebrospinal fluid to be able to do such a clear and well-defined distinguishing of different types of Alzheimer's.
Yeah,
Yes.
that's true. Now, what they applied this to some other cohorts as well, so some data that they have. And what I find really striking was that they could find these subtypes in other cohorts as well. So they couldn't know that. So they just applied that what they knew and find out that most of the subtypes can be found in any cohort, which is kind of really strengthening the method to say that we need to think about that Alzheimer's is this heterogeneous form, and that we need to identify actually the subtypes so we can actually help the people more in that way. So now I want to come to an end with this little journal club, of course. So what do you think are the limitations of the study?
Well, the authors already discussed a couple of the limitations that they saw themselves, and one of them is that they based their subtyping only on the biomarkers, right? Even though they do show a strong correlation with the pathology, they assumed that sometimes this might not be completely accurate, right? And as you had already pointed out, that they have their cohort where they did it very thoroughly. But I guess one has to go into other cohorts, which means other patient groups from different parts of the world, and to see whether this is not just something going on in the Netherlands. So this is one of the weak parts of the study that they identified themselves, and what they also didn't have access to is whether these subtypes that they have identified did react different to treatments. And this is something that this study really wants to support, that different subtypes of the disease might need different treatment. Yeah. As you said, personalized medicine.
For instance, in your case, subtype 1 might benefit by inhibiting the dysfunctional
TREM2, yeah.
TREM2.
Or even replacing it.
Replacing it with CRISPR-Cas.
For instance.
Maybe also for the other subtype that had the blood-brain barrier subtype. I read that you could do antibody-based stuff, but the problem is that in these patients, it could be, since they are microbleeding, you could force even more bleedings. So, but this is kind of important information, right? So if you have a patient in front of you that maybe has especially that subtype, for instance, so if this is really going to be later on in the guidelines, you know that a specific treatment will not help. It will worsen the conditions, right?
Yes. But maybe we haven't made that clear at any point so far. At the moment, there is not really a treatment that treats and eliminates the disease.
Sure.
We have not discussed that.
True.
Just to make
you are right,
that no one is confused, right? But of course, in clinical trials, one has been trying over the last couple of years and tested different approaches, and one is the one applying antibodies against the plaques, for instance, so that they are marked, and then the immune system of the brain can remove them a lot better, right? So this is just one thing, but the journal club wasn't meant to investigate all the treatments, but just in case someone now thinks there is already treatments, there is a couple of things that one can try, clinical trials. There is some medication that increases for at least a while the level of some transmitters, and thereby supports a little bit more the mental health for a while. Yeah. But at this point, there is no treatment, unfortunately, for this really devastating
unfortunately
disease.
True. Yeah, and again, we are not doctors. We don't know how to do the treatment of a patient. We are just talking about the science behind it, and it's a medical thing. And I love if maybe a medical professional out there leaves a comment down and says, "Hey, that's cool." But I could imagine like helping the doctors if they know the subtype, like that would be something, if there would be a treatment at some point, that they can tailor it actually, because they were also really clear that we need personalized medicine for this.
Yes, I absolutely agree. From the data that they show, it's very convincing that there is not the Alzheimer's disease. There is a plethora of different subtypes. It might not stay with the 5, and that is one of the weaknesses that I see, not really of the study, but of the matter itself.
Yes.
As you explained, this very complicated, at least for me as a neurophysiologist, complicated technique to investigate the proteins, I know that in the next couple of years, the methods are developing further and they are even more sensitive. So maybe we sit here again in 2 or 3 years and we find a publication where they have identified even more subtypes, which makes it again complicated, right? And more complicated, and you even have to check out more values to decide which subtypes your patients have to be grouped in, right?
Yeah. And another thing that was not written in this paper, what might be also a limitation, you have to think how they did it. So from a method perspective, you cannot really say that because you don't do mass spec in that sense. But for me, I know it's really a lot of work to analyze the data. Sample preparation, I would say, is okay. But I imagine someone wants to implement this in a clinical setting. You need an expert who can run this machine, who can analyze it, because you cannot let it made a doctor. The doctor doesn't have time to do that. And if I imagine having that at Klinikum Großhadern, and no, I don't see it. So this is also a weakness that we have to maybe point out, that this kind of method is not copy-paste into it. Yeah, you cannot do it. So
it's not for everyday use at the moment.
Yes.
But things might change. We don't know.
Yes. So, of course, there are stuff developing. But these kind of complex data analysis, in the end, to really interpret them right, you need a lot of people, a good team that understands how mass specs work and so on. So this is also something that I tell people also, like, mass spec, yes, is cool, but we cannot apply everything on this, because in the end, you need someone who understands the data, right? So this is also like, so you will not have this test within the next 2 years in the, but I think it's cool to know that science is developing new methods or trying to analyze the fluids that we have so we can understand better what is going on in disease and in the healthy state.
Yeah. But analysis like this might also lead to that we find quite stable markers that are very specific for some or one of the subtypes, right? And then one wouldn't have to analyze all the proteins anymore. One could really
pinpoint
and look only for these specific markers.
Definitely.
So from what the data set that they deliver is a really good base for going on a hunt for something like that. Would you agree?
Completely. Because in the end, if someone else does a cohort in that similar way, he or she can actually compare the data from back then, right? And see, do we find more or less, or maybe other markers as well. So this is really like a treasure that they are making. And huge kudos to these people. So like, it's a big collaboration between many countries and many people, and I really appreciate that they are doing so cool science. I think we should wrap up and make a small take-home message. So what did you take from this study?
Well, for me as a neurophysiologist, again, it made it so clear how important analysis of the CSF can be, right? So, and the importance of the investigation of that body fluid.
Yeah.
That was one thing. I wasn't aware of that in this
context.
Yeah. I found this subtyping, because I do believe not only for Alzheimer's but for many other diseases which very often are very heterogeneous, the future of medicine will be this personalized medicine. Yeah. So I think a very important example is cancer treatment, right? So I find it really cool how well defined they were able to identify these subtypes. So that's really cool for me, which is a good base for developing personalized medicine.
Yeah, for me as well. So they had these 5 subtypes. They could pinpoint it down to either it's the immune system that is up or down regulated, that RNA has something to do with it, then the choroid plexus has also something to do with it, and in the end the blood-brain barrier actually also contributes to the AD disease, which might be not so straightforward, right? Because why should the blood-brain barrier do something with it? But makes in that context sense. Also correlating it to the genetic risk variants and also the MRI scan, so that you have also how is the atrophy going on in these patients. And they even looked at the average survival rate. So that was also really interesting, that I think it was subtype. So subtype 3 showed that it has an average survival rate of 5.6 years, whereas subtype 1 had the longest average survival time of 8.9 years. So this is also like kind of interesting to know that even the survival rate somehow correlates with each subtype.
Yeah, because the severity was...
the severity, yes.
different.
So, yeah, so that's the take-home message of the paper. And of course, it's not an invitation now that we say this should be implemented immediately as a method, right? But I think you got the essence of what does it mean and why the CSF is maybe cool to actually analyze, and what scientists are making out of it, and what kind of information can we get. And I'm looking forward to more of these really cool scientific papers that really take the biofluids from us and analyze it, right? And yeah, that's already it, guys. Thanks a lot. Thanks, Corinna, for coming and giving your expertise on this. It was a really nice talk. You know, I really enjoyed also our meeting where we were discussing about the paper already, and you gave me so many insights on the brain and how it's working. So yeah, we're gonna see us in the next episode. Let's see when it will be. So I cannot make claims here because I'm currently working a lot. But yeah, leave a like, subscribe, please. And please leave some comments, right? So if something was not clear, or if you want to know something more, just let me know. And looking forward, right? And big applause to Corinna once again. Thanks a lot. And see you. Bye.
Bye.